Hey, good morning or afternoon to you. Thanks for joining us for this presentation of Antibodies to Watch in 2023. I'm Jan Reichert, Chief Operating Officer of the Antibody Society, and I'm here today with three of my co-authors on the paper that forms the basis of this presentation. Alicia, would you continue with introductions?
Sure, thanks, Jan. I'm Alicia. I'm a postdoc at King's College London, and I primarily work on engineering the FC region of antibodies for better therapeutic function. And it's always really fun to go and do an Antibodies to Watch in 2023 because I get to see all the cool new engineering strategies that are coming out, which you will hear about later. So, Sylvia?
Hello, thank you, Alicia. I'm a senior postdoc in the Cancer Antibody and Immunity King's College London, together with Alicia. I work on antibody engineering and on the study of B-cell response in cutaneous melanoma. And as always, I'm very happy to be part of this Antibodies to Watch. It's very, very interesting editorial, and I look forward to guide you through this our work. And I will hand it over to Jyotsna.
Thank you, Sylvia. My name is Jyotsna. I head the antibody discovery group at Seismic Therapeutic, an AI machine learning immunology company. Our focus is on making therapies for autoimmune diseases and people suffering from elevated or inflamed immune systems. I really enjoyed working on Antibodies to Watch this year, and I will make this promise on air, which is I would love to work on this again because this was such a rewarding and fun experience, also working with Jan and the co-authors on this paper. Like everybody else said, I'm excited to walk you through all the upcoming trends and which antibodies you need to keep an eye on.
Thank you very much, all three of you. And Jyotsna, I'm pleased to hear that we didn't scare you away this year. The paper on which this presentation is based is 42 pages long. It was a challenge. So Antibodies to Watch in 2023 is the latest in an article series that started with Antibodies to Watch in 2010. Each paper provides a snapshot of the late-stage commercial pipeline of antibody therapeutics as of the fall of the year prior to the year in the title. Thus, Antibodies to Watch in 2023 was written in the fall of 2022. However, we have updated this presentation to reflect events that occurred during December 2022 and early January 2023. Okay, so the first topic will be trends in approvals of antibody therapeutics in the US or EU. In all cases, we are referring to first approvals. Any subsequent approvals for supplemental indications for antibodies that were previously on or that are on the market that is previously approved, those are not included in the totals that we'll talk about. So I'll then briefly review the antibody therapeutics that are first approved, so give you the specifics of the antibodies first approved in the US or EU in 2022. And we have had one already in 2023, so I'll mention that one also. I'll also briefly discuss the antibody therapeutics that are currently in regulatory review in the US or EU. And so these are antibodies to watch for approvals in 2023. The second topic will be first approvals of antibody therapeutics in the rest of the world. So what happened outside of the US or EU in 2022 and those in review in the rest of the world. So again, antibodies that may be approved outside of the US or EU in 2023. And you'll notice all of the activity is in China and Japan. Next, Jyotsna, Alicia, and Sylvia will take over and they'll discuss trends in the late stage development of antibody therapeutics, specifically focusing on the types of molecules that are being developed. I'll then give you our forecast for antibodies to watch for possible transition into regulatory review. Josna will then come back and talk about trends in the development of bispecifics, because you'll be hearing the word bispecific many times in this presentation. Okay, so for our first topic, trends, first the trends in first approvals and then the specifics. So this graph shows the annual number of first approvals for innovative recombinant antibody therapeutics that were granted in the US or EU each year since 1997, which was the first year in which 2 or more were approved. So I do want to emphasize we are talking about innovative molecules, so no biosimilar approvals are included here. And we are talking about recombinant, so just to make it clear, we do not follow polyclonal mixtures of antibodies unless they're recombinant. Then we do. There's always exceptions to the rules. So you'll notice the pattern of approvals between 1997 and 2013 is quite different from the pattern that started in 2014. Between 1997 and 2013, with the exception of 2009, you notice the number of annual approvals was in the range of 0 to 4, so reasonably low. But that pattern did change in 2014, running through to 2022, when the number increased to between 6 and 13 every single year. And that's a consequence of the larger number of antibodies in late-stage clinical studies. And we will be telling you all about that relatively soon. So in 2022, we did match the record number, which was 13. That record had previously been set in 2018 and matched again in 2021. 2022, we see that 13 number again. In looking forward to 2023, I'm going to be a little bit conservative with my estimate and put it at 11 for now, and I'll give you the specifics of those. 11 is certainly achievable based on the data that we have now, and more are certainly possible, and we'll tell you about that. This table and the next one list the 13 antibodies that were granted a first approval in 2022 in the US or EU. So this table shows the 7 that are for non-cancer indications. This presentation can be downloaded from the viewer, so I'm not going to go over every single row of every single table because there's a lot of details here. The information that I think is most notable is shown in the bold red text. So I would direct you towards that bold red text for the discussion moving forward and continuing into the presentations. So as I mentioned at the outset, you will see that word bispecific many, many times. And it starts out the presentation here because faricimab, which is a bispecific antibody derived from Roche's CrossMab platform, was the first approval in 2022. We also had an approval in the EU for a mixture of 2 antibodies, both of which target SARS-CoV-2. This particular product was not approved in the US. It was granted an emergency use authorization, though. But like similar products, that EUA has become a little bit in question. Previously approved anti-SARS-CoV-2 products that had gotten EUAs in the US have been revoked. So that EUA has been revoked. Those are no longer available. Evusheld looks like it might be trending in that direction because there are now some questions about whether it appropriately neutralizes or suitably neutralizes Omicron variants. But as of this morning, when I checked, that EUA was still valid. We'll see how it goes in the rest of 2023. The next notable antibody is Beyfortus, and that is an antibody that was approved in the EU for prevention of RSV infection. So RSV is respiratory syncytial virus that is a rather pernicious infection that tends to be gotten by neonates infants and elderly adults. The approval in Europe did allow it to be used for the current RSV season. A BLA has been submitted in the US, but of course that will not make the product immediately available. We do look forward, hopefully, for that being on the market for the 2023-2024 RSV season. Teclistamab is another notable approval. It is for the delay of the onset of type 3, type 1 diabetes, and that approval was in the US. We're not aware yet that an application has been, has been filed in Europe. So teclistamab, so again, like Beyfortus, it is not a traditional antibody in being a treatment. It maintains function by attenuating the T cells that mediate pancreatic beta cell death. And so it interferes with the progress of the disease. In looking at the next set of antibodies, so this is the 6 of the 13 antibodies that were approved for cancer in 2022. So of the 6, 3 are bispecific T cell engagers. Kimmtrak's GP100 binding site is derived from a T cell receptor rather than from an antibody gene. So here I've referred to it as an immunoconjugate. Another bispecific granted an approval in 2022 binds CD20 and CD3. The third one here binds BCMA and CD3. So all 3 of these are T cell engagers. I've highlighted in red the targets GP100, as well as the combination, of course, GP100 and CD3, LAG3, the combination CD20 and CD3, and folate receptor, and LAG3 and folate receptor alpha. So all of these targets are unique compared to targets for the antibodies that were previously on the market. So these antibodies do have unique mechanisms of action compared to previously approved products. So finally, we do want to point out the one ADC that was approved, and that is ImmunoGen's molecule for ovarian cancer. And moving into 2023, of the 11 that we hope will be approved in 2023, or have a good, warm, fuzzy feeling about that, one of them has already been approved. So that is lecanemab, which targets amyloid beta, and it is for Alzheimer's disease. And we look forward to having more antibody therapeutics join this table throughout the year. Now, I'll talk to you about which ones those might be. The antibodies that are in a first regulatory review in either the US or the EU, and thus hopefully queued up for approval in 2023. So here, this table and the next one does list those antibodies for which a marketing application is already under review in either the US or the EU. So I'll start with the 9 that are either in review in both of those regions, which is most of them, 8 of the 9, and the 1 that is in EU review only. So antibody therapeutics are typically approved in both regions eventually, but there can be a lag. Sometimes it's a substantial lag, sometimes it's only a few days, but there can be a lag between the dates of the marketing application submissions to EMA and FDA. So of course, therefore, that plays out as a difference in, or a lag in, the approvals in the 2 different regions. So we do keep track of both regions and watch what goes on with them. So here it's clear the pandemic is having an effect on the agencies still. In the case of toripalimab, the BLA is under review, but it had a PDUFA date in the end of December. The FDA had travel restrictions, though, and therefore the decision was delayed. So we kind of were expecting, hoping for, because that would have put us over the top with the number of approvals. It would have been 14. That would have been a record. I would have been all super excited about that. But unfortunately, there was that delay, so we're still waiting. We do have PDUFA dates for 3 of these additional molecules here. One is an ADC Two are bispecific antibodies. So both of those bispecifics are T-cell engagers, and you'll notice they both target CD20 and CD3. So these are fast followers of the antibody that targeted CD20 and CD3 that I just pointed out to you as an approval in 2022. Another bispecific in this group targets G protein coupled receptor, family C, group 5D, and CD3. So again, a T-cell engager. Lots of those getting to regulatory review and onto the market. In the last row, I've highlighted this molecule where it is undergoing a second review cycle, and I will pick up on that theme in the next slide. First, though, I will point out, well, first let me point out that these 7 antibodies are in review only in the US. We are not aware of an application with EMA yet. I looked this morning, trying to keep you completely up to date with what's going on. I did look this morning as to whether EMA had issued their list for January, but it is not as yet online. So very, very nicely, EMA does release a list of candidates that are undergoing review, and that is put out monthly. So we do have that as a check on information that we're getting from the companies, which is really the only other credible source of information. FDA does not do that. That is not something that they do, unfortunately. So we do have to rely on company information, and sometimes the companies are not entirely transparent or timely in releasing this kind of information. So we do our best, but we can't guarantee that we're up to date every single second. It really is up to the companies to put the information out there. So we do have 2, and these are 2 that I did include amongst the 11 where it is a first review cycle. So one of them is an anti-amyloid beta antibody for Alzheimer's disease. So we do hope to see that approved in 2023. But you'll notice 4 of them are on second review cycles. That sort of thing used to be quite rare, but now it's a bit more common. It was quite rare for this number of products to ever be in regulatory review. So it's really a different expectation now for what's going to happen with antibody therapeutics in regulatory review. So just to refresh your memory or to let you know how this works, when BLAs are submitted to the FDA has a certain time period in which to review it and take an action. That action does not have to be an approval. It could be a list of things that they didn't like about the application and things that the company needs to address. At that point, though, the BLA is still considered under review. So I will continue to list it as in review, but I'll make a note that it's in a second cycle. So what happens next is that the company receives likely a complete response letter from the FDA. Then the BLA goes into what's called the sponsor response time. And in that period, they need to address the issues that FDA brought up. And if they address those appropriately, a BLA can be resubmitted. FDA will look at it again. And in theory, everything's been done perfectly and it's granted an approval. Practically speaking, though, that does not have to be the case. The BLAs can go through an infinite number of cycles. One hope that doesn't happen, but I have seen with small molecules 6 and 7 different review cycles. So we're again hoping that the responses are suitable. But in the best case scenario, this second review cycle will definitely lengthen the review period. It will just take longer, but occasionally I do have to point out it can derail a BLA entirely. And that can happen when the sponsor cannot address those issues. And you'll notice here with omburtamab, the company sponsoring this particular molecule is Y-mAb. They have already noted that this asset is being deprioritized. So it's likely the BLA on this will be withdrawn. We also point out pinpulimab, which is an antibody where the company said, actually, no, quite some time ago, that it was undergoing a real-time oncology review, but then we never heard another word about it. So it's again, an issue where if the company does not provide additional information, then we just simply have to kind of put it in limbo and point out it's something where we're not certain what's going on with this molecule. Now, moving on to hopefully a more positive topic. I'll briefly review what's going on in the rest of the world. As I mentioned, this was mostly, well, entirely China and Japan. So China is really coming up in terms of being the country really of some first approvals as well as first regulatory submissions. So I note here 7 approvals that occurred first outside of the US or EU. 5 of them were in China and 2 of them were in Japan. Both of them were bispecific. So again, that word bispecifics. One of the bispecifics targets 2 immune checkpoints, and that's CD1 and CTLA4. And the second one is the first approval in Japan of a bispecific nanobody. Now, looking at the antibodies that are in regulatory review. So if an application is not filed somewhere else, or it is filed and that regulatory agency doesn't move as quickly as the one in China, then we would expect a first marketing approval to occur in either China and Japan for this particular set of molecules. And I will point out crovalimab in particular because the application was filed by Roche. So this is a major multinational organization that has selected China potentially, again, if the timing works out, as the first market for their antibody. So very exciting to see all of this activity happening in areas outside of the US or EU. And with that, we will transition to the trends in late-stage development of antibody therapeutics, and I will hand things over to Jyothna.
Thanks, Jan. So now we'll talk a little bit about the trends that we noticed while writing this review about the late-stage antibody therapeutics. So here we are presenting to you all antibodies in late-stage studies over time. The bar graphs are split as red for cancer therapies and blue for non-cancerous therapeutics. And as you can see, as it was already shown earlier, over time, these numbers are going up with the estimate for 2023 as of October last year being roughly evenly split to about 70 and 70 across both cancerous and non-cancerous therapies. And this is, as we expect or forecast, this is only going to get more over the coming years, which is good news for patients waiting for therapies. Moving on to the late stage pipeline, the formats over time. So what we did was we took all these numbers and broke it down over formats. So in A is non-cancerous therapies and B is cancer therapies. So as you can see, so we'll look at the cancer therapies first, that there are many flavors than just the standard full-length IgGs or MABs, which is represented here in the light blue color. But then you can see over time, the ADCs or the antibody drug conjugates, as well as the immunoconjugates, are increasing late stage in the late stage pipeline over time. And then also there are a large number of, you guessed it, bispecifics, also now in the late stage therapies. However, it's a little bit more on the full-length antibodies in the non-cancer field. But as you can see, in the past few years, the past 4 years, for example, there is space, and you can see the different formats are being brought into late-stage pipelines here. And we expect that a lot more to be coming in the next few years. Now I'll hand it over to Sylvia for the next.
I think I'll do the next one, but thanks. So when we saw this movement heading towards more novel antibody formats in the late stage clinical trials, we also wanted to highlight the diversity of the formats being used and the different sort of engineering techniques that have been applied to the design of antibodies to enhance therapeutic effects. So in the top two graphs, we separated out format based on IgG subclass. So if they're a full-length antibody, so this includes bispecific, ADCs, immunoconjugates, we classified them whether they had an IgG1, IgG2, or IgG4 backbone, or if they're a mixture of multiple antibodies, or if they're an antibody fragment, which will be classified as a broad category, including SESBs, VHH homodimers, Fabs, basically any non-full-length antibody. So as you can see, the majority of antibodies for both non-cancer and cancer indications are based on a full-length IgG1 backbone, while approximately a third of antibodies in both indications are formatted onto the less immunoactive subclasses IgG2 or IgG4. This is often done to reduce immune engagement when an immune response would be unwanted or detrimental, such as for checkpoint inhibitors for cancer or for blocking ligand receptor interactions in non-cancer indications. While the details of all the formats have not yet been divulged for the late stage clinical trial antibodies, we know that there's at least 8 antibody fragments in the late stages, 3 for non-cancer indications and 5 for cancer indications. And we know that antibody fragments may have some beneficial properties over full-length mAbs, such as better tissue penetration, access to difficult to reach targets, and minimal undesirable immune activation due to the lack of Fc domain. However, one of the main drawbacks of these fragments is the greatly reduced half-life. But we are seeing different engineering strategies to improve this feature, such as pegylation. So when looking at all the engineering approaches for each molecule, our data shows that over 2/3 of them have been engineered to improve their therapeutic effect in some way. One of these strategies, as we've heard many times, is bispecific assembly, and you'll hear more about the bispecifics later. But another way is to engineer the Fc domain to have differential binding to different Fc receptors or to FcRn. So engineering the Fc domain to increase binding to FcRn, and therefore increase serum half-life, is getting more commonly seen in non-cancer indications, especially in antibodies against infectious diseases caused by agents such as SARS-CoV-2 or RSV. We have 7 antibodies which have been engineered for increased half-life for non-cancer indications, compared to just 2 in cancer indications, both of which are for checkpoint inhibitors. Other engineering strategies that include increased affinity for Fc receptors, such as improving ADCC via afucosylation, is commonly used in cancer indications as an increased immune response is desirable. However, some antibodies have also been engineered for increased affinity for Fc receptors for a different purpose, for the non-cancer indications. An example of this approach includes sweeping antibodies, which have been engineered to have enhanced Fc-gamma2b and FcRn binding to rapidly and potently clear out antigen of the circulation. And another example is increased neutralization of lipid polysaccharide signaling via toll-like receptor 4 by enhanced Fc-gamma2a binding. We also have a number of ADCs and immunoconjugates, which use the specificity of antibodies to deliver other payloads directly to the target. So these payloads can be toxic drugs, cytokines, or cytokine binding domains. And this form of engineering is much more common for cancer indications compared to non-cancer indications. As I mentioned before, similar to the use of IgG2 and IgG4 as a backbone compared to the more immunoactive IgG1, the reduction of immune effector function can be beneficial in both cancer and non-cancer indications. So, however, instead of using IgG2 or IgG4 backbone, which can still have some immune function, there are many antibodies which now reduce immune function by introduction of point mutations into the Fc domain, which is what we've called Fc silent mutations. And this is one of the more commonly used engineering approaches. So I'll now hand it over to Sylvia, who will talk about the mechanism of action of some of the antibodies in the late stage.
Thank you, Alicia. So we have assessed the main, let me change again. So we have assessed the main mechanism of action of the antibodies in late stage studies. And here we represented the data as Euler diagrams in order to represent also the overlaps when antibodies have multiple mechanisms of action. We have on the left-hand side the mechanism of action of antibodies for non-cancer indication. And the majority of these antibodies are blocking or neutralizing antibodies. We also have 11 antibodies with antigen clearance properties, 4 with cell depletion properties, and 1 agonistic antibody. The agonistic antibody is MIM8, an antibody which is a bispecific and is designed to target factor 9 and factor 10 for the treatment of hemophilia. And then we have, as you can see, antibodies with a dual mechanism of action. We have 2 antibodies with both blocking and cell depletion properties. One of these antibodies target T cells via OX40, and another one target B cells binding to BAFF receptor. Then we have 4 antibodies with both blocking and antigen clearance properties. And these are antibodies targeting triglyceride, IgE, amyloid, and tau. Then on the right, we have the mechanism of action of antibodies for cancer indication. And here we can appreciate a greater diversity in the mechanism of action. However, we can still see that the majority of these mechanisms of action are blocking. But however, we could see also antibodies designed for payload delivery. We have 16 antibodies for payload delivery, 11 antibodies with antigen clearance properties, 7 antibodies with cell depletion properties like complement-dependent cytotoxicity, antibody-dependent cytotoxicity, and antibody-dependent cell phagocytosis. Then we have 7 bispecific cell engagers and 2 agonistic antibodies. Of the blocking antibodies, the majority, 31 of them, are immunomodulatory. Then we have 9 antibodies targeting the tumor and 3 antibodies targeting the tumor microenvironment. Of the ones that are designed for payload delivery, which are 16, 13 of them are antibody-drug conjugates. Of these, speaking about warhead types, we have 6 antibodies which are antitubulin, 4 which carry DNA binding agents, and 3 carrying topoisomerase 1 inhibitors. Then of the cell engagers, we have 6 antibodies which have been designed to recruit T cells, and 1 designed to recruit natural killer cells via CD16A. Also, all of these antibodies, except 1, which targets delta ligand like 3, DLL3, which is expressed on solid tumors. All the others are designed for hematological malignancies. Then the 2 antibodies which are agonistic are 1 antibodies which target death receptor 5 for the killing of tumor cells, and 1 antibody which target OX40 for the activation of T cells. And then here as well, we have antibodies with a dual mechanism of action. We have antibodies with both cell depletion properties and blocking properties. We have antibodies which are antibody-drug conjugates, but which also have the ability to block receptor signaling. One of them is an anti-c-Met, and the other one is an anti-ROR1. And then we have 7 antibodies with both blocking and antigen clearance properties. Of these, 1 is targeting CD73, Another one is a mixture of antibodies which target non-overlapping epitopes of EGFR. And the other 5 are bispecific antibodies. Two of them target both PD-L1 and TGF-beta. And the others target VEGF and either DLL3, 2 of them, or PD-1. And then we have one antibody, zanidatamab, which is an anti-HER2 antibody, which has a triple mechanism of action. So it's able to block receptor signaling, to mediate antigen internalization and clearance, and to trigger ADCC. And now I will hand it over to Jen.
I think Jan's having a little bit of technical difficulties for the moment. So we can just move along. So I'll just keep going for now. So we're going to also have a look for antibodies to watch for possible transition into regulatory review for 2023. So we have 5 of them at the moment, which are potentially going into regulatory review or it's anticipated in 2023 for the non-cancer. We have 3 of them that are IgG4s and 2 that are IgG1s. And the one that we've got highlighted here is an antibody biopolymer conjugate, which is very cool. So we are going to look for all of these, and hopefully these will all go into regulatory review this year. They're looking forward. We also have 6 anticipated for cancer. Probably got more for cancer actually, but 6 in this graph. And highlighted here, we have an antibody tiragolumab against TIGIT and zolbetuximab against claudin 18.2, both for cancer. Oh, Jan's back. You can take over from now.
I seem to still be on mute, though.
No, we can hear you.
Oh, here we go. Okay. Yeah.
I tried to hold that.
Yeah. Yeah. Somehow I was getting a message to ask to be unmuted, and it wouldn't do it. So thanks for hopping in there and getting us to this point. And now I'll try to get us to where we're going next. So I had taken the antibodies that are in regulatory review, and for the ones for cancer, split them up into 3 different groups. One of them being standard canonical antibodies that may be remarkable in the antigen in particular. So the TIGIT and claudin 18.2, those are antigens that are unique amongst the ones that are in or that are on the market. So now we're going to flip forward to the next set, which are the immunoconjugates. So here we have 4 different immunoconjugates, and these in particular include one that's radio-labeled. So that includes the isotope iodine 131. And we also have 3 different antibody drug conjugates. So these are antibodies that are conjugated to small molecule drugs. Next, we have our bispecifics. Yay, more bispecifics. So this is a good segue into the section on trends in bispecific antibody therapeutics development. Here I've included 5 that have the marketing application submitted. And so therefore these are potential approvals in 2023. 3 of them are T-cell engagers. One targets 2 epitopes on HER2. And I want to point out particularly, though, the erfonrilimab which does have quite an unusual format compared to the others. And also to point out that in this case where I've noted that it's an NDA, this application we're expecting to be filed in China. May follow on with an application later in the US or the EU, but first application looks like it's going to be directed to China. With that, I will not mute myself this time, but I'll hand it over to Jasna.
Thanks, Jen. So we'll, quickly wrap up here, taking you guys over the trends in bispecific antibody development over the years. And so you can see here that while there were one and few bispecific approvals in the past few years, you can see this sudden steep increase in activity and approvals in the past 2 years, 21 and 22, and we expect a few more coming out in 20, which are projections as of last year when we were writing this review. So while this is still like, if you compare to the rest of the numbers, a small number, but for the bispecific field, it's huge. And also, I think this is with the new crossbody, or no longer new crossbody, cross-mab and duobody technology, we expect to see a lot more coming into approvals and late stages in the future. And then I just wanted to also talk a little bit about the 8 bispecifics that were approved in 2022, which with 3 more, there is the number 3 there on that graph, it's hard to see, in review currently. And a lot more in phase 3, which is, 18 is a huge number for bispecifics. And this is really fueled, as you can see here, by the large number of molecules getting into the phase 1 and phase 2 trials. So I would say watch the space. The bispecifics are going to become a big players in, and you'll see and hear a lot more of these. I think another thing we wanted to just mention out here was that of these bispecifics, 1/3 of them were T-cell engagers, with about 5% of those in late stages of development, and of the 2/3 that are not T-cell engagers, about 6% of those were in late stages of development. With that, I will hand it back to Jen to add any finishing.
Thank you very much. And here I am. I didn't disappear this time. So I will just wrap up here with some key messages that hope were fairly clear from our presentation. We're very excited that we could match the historical record of 13 approvals in either the US or the EU in 2022. It's remotely possible that we could match or potentially exceed that number of approvals in 2023, but it would rely on some of the molecules that we were just discussing that are in late stage clinical studies now to already get into regulatory review, receive, especially regulatory review in the US, because if an application is filed by the end of June in 2023, it receives a priority review, and it's a really good application that the FDA moves on with an approval as the first action, then it's technically possible for that all to happen in 2023. So I can never say for sure, well, we've got X number in review already, so that's what we've got for possible approvals in the next year, because there's always that little wild card of applications that could be submitted and just like zip right through very quickly. On a rare occasion, FDA will actually take action before the PDUFA date. It has happened with some of the early checkpoint inhibitors. I believe some of them, at least one, was approved upwards of 2 months before the PDUFA date. So you never know. You just have to kind of keep track of what's going on all the time. I did point out, though, that an entry into regulatory review doesn't necessarily mean you will zip through all that quickly. We have a large number in review. So again, now for the past couple of years, it's been in the range of 15 to sometimes over 20 antibodies in review all at the same time, which is highly unusual. And so I suppose not unexpected that a few of those BLAs will start to have some problems. We do have a number that are in second review cycles. That does slow things down. So the number of approvals is not quite keeping pace with that rate of entry into regulatory review. But again, we'll keep track of it. We'll let you know how it's going. Definitely that late stage pipeline is far exceeding my expectations. It's growing at a crazy rate. But again, another thing we'll keep track of. The antibodies to watch papers may start, getting into the 50 and 60 pages long, which will start being rather excessive. But nevertheless, we're here to keep you informed, so we'll do it. We do see lots of activity in that bispecific space, a lot of bispecifics in early stage clinical studies. We do hope more of those will transition forwards and get onto the market relatively soon, because any number of them certainly have unique mechanisms of action and hopefully have mechanisms of action that will be of benefits to patients that are currently in need and meeting some of that unmet medical need that's out there. Finally, I direct you to the Antibodies to Watch in 2023 paper, which was published in MABS in early December and available for you to download, all 42 pages of it. Finally, I do want to acknowledge our other co-author, Ellen Kaplan. She has been wonderful, absolutely wonderful, going back to Antibodies to Watch in 2018. So she has been the longest running co-author on Antibodies to Watch, and we could not have done it without her assistance. I also, of course, want to thank the Antibody Society and all of their sponsors for their support. Just want to take a second here also to encourage you to join the Antibody Society, especially if you like this kind of business intelligence content. We do focus on that commercial antibody therapeutic pipeline and what's going on in general with all sorts of antibody news. If you're not already following us on LinkedIn, you'll get lots of information just by following us on LinkedIn. We do send emails to members, and again, it's all sorts of content, business deals and acquisitions, financing, things the regulatory agencies are doing in terms of providing various designations to antibody therapeutics, including orphan drug, fast track, breakthrough therapy designations, EMA's prime designation. We do like to track firsts on everything. So the first time an antibody enters clinical studies, and the more advanced studies, a first phase 2, a first phase 3. So we always make note of that. Certainly any marketing application submissions and approvals, but also it's not all positive news. So we do also let you know about withdrawals and terminations that may occur. And of course, this is all grist for our Antibodies to Watch mill. There will certainly be an Antibodies to Watch in 2024, so watch for that later in the year. We do, of course, also offer the late-stage pipeline information right on the website. So we have the late-stage pipeline as well as antibodies and regulatory review and approved in 2 different tables that can be downloaded from the Antibody Society's website. And if that was not enough, we also track, of course, the entire pipeline so that we know what's going on. That data we do provide to our corporate sponsors. In the last couple of slides, I will specifically thank the corporate sponsors of the Antibody Society for their financial support. I do have the logos of the companies here. They are in alphabetical order. So take a second and just look to see if your company's logo is represented here. And the reason for having a look would be if you're not already registered as an Antibody Society member, I would encourage you to do so because your company is already paid for it. We have a membership level that is specifically for corporate sponsor employees. So you can go to the website if you see your company's logo here and join for free. And we also have our adaptive immune receptor repertoire community. If you're interested in that topic, I would encourage you to get involved with that group. They do have a separate meeting that does raise its own money for in this particular case, it was a meeting in May, and we thank those companies also for their financial support. With that, I thank you kindly for your attention. We're happy to answer the questions that have come in, and I've provided the specific places on the website where you can find those tables. They are in the web resources section. So with that, we'll now start taking a look at all of these questions coming in.
Okay, I can start reading out the questions, and I think we should start with one that's the classic theme of this, is about bispecifics. So we have a question if there's a lot of bispecifics, and which technologies are used the most to facilitate correct structure, avoiding mismatches, and are we aware of any manufacturing challenges, such as stability compared to standard MABs?
So I would say for that one, we might want to hop to Jatna, who is our bispecifics expert, to perhaps give some background on that.
Okay, I was just making sure I was unmuted. So obviously there are always manufacturing challenges with bispecifics because you need to make sure the right chains, the heavy and light chains are paired, as well as if it depends on the formats that you choose. So there is a whole slew of challenges that come with the format that you choose. So most people along the way try to I guess, de-risk a lot of these going in, putting a lot of thought into formats and, the cross-map technology, all of these account for those sorts of mismatch light chains, as well as you will see a lot of people have come out with common light chains. So you could try to discover antibodies against your targets using common light chain animals that have been genetically modified to have the exact same light chain, so you don't have this light chain mispairing and mismatch happening. So a lot of strategies there to avoid, but definitely manufacturing can be challenging, and that's something most people are aware of and take into consideration when they're designing their therapeutic, the format.
Also, just as a small shout out back again to the Antibody Society therapeutic, like the late stage clinical trial tables and the proof tables. We also do have, for where it's known, which mutations and engineering strategies have been used to develop each of these bispecifics. So you can go on the tables and find out how each one has been produced, pretty much. Yeah, so just a shout out for that. Another one says, do we have any trends on single domain antibodies coming through the pipeline?
So for that, I would definitely encourage you, Alicia, if you specifically noticed anything in the late stage pipeline. In the early stage pipeline, I guess what I would say is that the single domain antibodies have actually been around for quite some time. And the challenge with them is to, well, frankly, find a purpose for them. If they're the right kind of antibody to meet a particular need, then they would be developed for that. But it's usually the default option to start with a canonical antibody. And it's when you can't do something with a canonical antibody that you would move to something else. And certainly we see that in the cancer space, the canonical antibody, and that's why you see so many of these different formats, so many of these different approaches, is that a standard canonical antibody can't necessarily do the job in cancer. So you want the ADCs, you want the, potentially the radiolabeled one that I mentioned. There aren't so many radiolabeled ones actually in the pipeline, but the antibody drug conjugates, the bispecific antibodies, again, adding functionality to them that you can't achieve with a canonical antibody. And so then you have to ask the question then, well, with the single domain of antibodies, they do have a purpose in some niche applications, but we, at least from my perspective, what I've seen, it hasn't led to a big increase in them in the pipeline. Alicia, would you agree with that?
Yeah, so in the late stage clinical pipeline, I don't think we had any single domain antibodies on their own. The only one we had was a single domain antibody that was fused to an FC to kind of make a heavy chain by itself. But as far as I'm aware, in the late stage, at least what we were looking at, no single domains by themselves. No. Someone's just said that they really love the Antibodies to Watch series and wants to thank us all for the effort. So thank you very much.
Yeah, I have to say, though, that's the first time I've ever seen anybody abbreviate it as A2W. It's like, yeah, that's really good. I like that. I like that. I'll pick up on that and use that myself. Thank you.
We've got talking about the new regulatory law saying there's no animal studies for FDA approval needed anymore. And what do we think about that and how it's going to affect the antibody market?
Well, first, I think FDA has their reasons for doing things. And if they're eliminating the need for specific animal studies, I think that's to the better. If it's unnecessary to use animals in these preclinical assessments, then definitely not. If you can get sufficient information from other sources, then why are you using animals to begin with? Because it's pretty well known that in some cases those animal study results really don't indicate much of anything. They don't help you when you're moving into humans, because the humans are not the same as the mice or the rabbits or even the cynos. So I think that's a very positive thing that they're getting rid of some of these old requirements for animal studies. I don't see how that will affect the antibody market. I don't see that as a negative at all.
Wonderful. And similar sort of question is it to talk a bit about the shortening regulatory pathways seeing in drug approvals for the FDA and EMA.
I'm not aware of any recent ones. I'm aware of all of the ones that have been around for quite some time. The misnomer really of the accelerated approvals is it doesn't really accelerate anything. That's really a reference to the fact that you can use a surrogate endpoint. So it may shorten clinical studies to a reasonable period compared to infinity with some of these. If you really had to look exclusively at, say, for example, at patient survival in some of the cancer studies, it would take a very, 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 very long time potentially. So I do, that is intended to make it more reasonable to get these molecules through the clinical development period and to accumulate sufficient information for FDA to assess that. But again, as is well known now, recently, the FDA is emphasizing that you need to have those confirmatory studies already started, or at least have a really good agreement with the FDA about how you're going to do it. But I think their preference is to have that already started, because they don't want to have molecules that were approved under the accelerated mechanism remaining on the market when in fact you do not get confirmation from a follow-on clinical studies. So other than that, again, the regulatory pathways exist to facilitate the process. It doesn't necessarily shorten the regulatory pathway, though.
Thank you. Two sort of similar questions. And the first is, do we document the origin of the antibody, whether it comes from animal or human derived or in vitro? And what is the most popular development platform for newer antibodies such as phage display? Yeah, so the short answer is we don't document the origin of the antibody, or at least we haven't, whether it's come from in vitro phage display or whether it's come from other sources for all of these, as it's a lot of work to go back and find every single origin of every single antibody that's coming through. It is probably possible if there's a big demand for it, but as of yet, it is not part of our tables that we have developed, that where it originally came from.
It is probably relatively straightforward for the late stage pipeline. It's not possible for the entire pipeline because especially for the ones in phase 1, they're just so limited amount of information available. But to a certain extent, though, it can be helpful in terms of some information, but it doesn't really help all that much, I don't think, because usually other things have been done to the molecule. So you can't necessarily say, well, this was successful or it was not successful, based solely on the fact that it's from this phage display or it's from a transgenic mouse. That's too simplistic.
And a question back on the bispecifics. On the CD3 bispecifics, are these mostly OKT3 or SP3/4 derived, or do they have novel CDRs? Anybody know? I don't.
Yeah, that one that may be a bit too specific. We could probably look it up later.
Possible to find, yes, but off the top of my head.
Drop us an email about that.
Yeah. So in terms of administration, are we seeing any trends on subcut administrations of MABs?
Yeah, I think especially for the antibodies that are for chronic indications, there's a real trend towards subcutaneous administration because it just makes it so much easier. You have a better patient compliance. You have better patient acceptance. It's better for the patient. It's better for caregivers. It's better for everybody if you can give it by sub-Q administration. But of course, it depends a little bit on the molecule because typically to give it sub-Q, you're dosing small amounts, then you need a high concentration. And so that's when your issues associated with self-association of the molecules, viscosity, all those sorts of things come into play. But definitely if you can do a sub-Q administration, that's desirable.
There's another question on targeted radionuclide therapy, isotopes conjugated to monoclonal antibodies. Do we have any data on that?
Well,
we have one, don't we?
Yeah, we report 2 radioimmunoconjugates. I think there are the details in the supplementary table of the antibodies to watch.
Using an antibody in a radio label form, the idea of that is very, very old. It goes back to the 1980s. The idea is not unique, certainly, and people have worked on it for now decades, and there's relatively few of them still. Some of that has to do with, especially in the U.S., the way the medical profession is set up, where the radio-labeled antibodies they require special handling. They can't just be administered by the usual oncology doctor. They need to be sent off to a special section of the hospitals. And in some cases, I have gotten the impression from others that there can be sort of like turf battles over the particular patients and whether the patients would do better on drugs that aren't radio labeled, don't have that additional complexity of dealing with a radioactive isotope. So it's been a hard sell in the U.S., maybe a little less so in Europe, but there are definitely barriers to the development of the radio-labeled antibodies, at least in the U.S., and they've never been a large portion of the pipeline. They aren't a large portion of the pipeline now.
Thank you. There's another question on the new proposed nomenclature for antibodies and when that's going to be started to be used. So I think that's the one that's not the MAB at the end, but all those new.
That is not terribly new. That there's at least one or maybe even more WHO lists that include those types of antibodies, the FUSPs and the BARTs, and I forget how many. There's at least 4 endings, I think, that are now relevant. It does make it more difficult for us because you can't just do a generic search on MAB and find everything that's MAB when you go to those WHO lists. You now have to search on all of those other endings and then sort out what the heck that thing is. So it adds a little bit of extra work, but it is already being used. There are now multiple antibodies that have those types of endings now. And yes, indeed, we are capturing all of that data.
Wonderful. I think we have potentially an answer to our previous CD3 question. As someone's just said, it's the SB34s or the Amgen SB34 likes. So thank you for that. And then there's another question on a very specific question on working on the generation of bispecific targeting human cytokine, and about whether it is better for an in vivo animal model to use a surrogate antibody or a transgenic model which expresses the human cytokine, which would be better.
Josna, I have to ask you to weigh in on that one, because that is far beyond my ability to answer. Any thoughts on the matter? Could be she has the same.
Okay. I mean, this we need more specifics. I don't know how to answer this. I mean, if it is truly a human cytokine you're targeting, maybe using the transgenic animal expressing the human cytokine, assuming everything else would be replicative of what you would expect in the human. So it's a hard question to answer straight up like this. So that would be my suggestion, but that doesn't mean that's the thing you should be doing. I'm sure you're privy of more information than me, so you should make a decision that would work well with your strategy.
Yes, the opinions offered here are those of the authors of Antibodies to Watch in 2023 and do not represent the views of our particular institutions or of the antibody itself, and we are not liable for any use of the material. Standard disclaimer.
I think that's all of our questions.
All right. Well, thank you very much. First, definitely in concluding here, thank you goes to my co-authors for all of their work on Antibodies to Watch in 2023. And I hope to have you back for Antibodies to Watch in 2024. And also thank you so much for being here to provide your insights and to help answer the questions relating to Antibodies to Watch in 2023. And I thank, of course, all of our attendees for joining the webinar today. An on-demand version will be available in a few days. I'll send a link to it to everybody who registered. Please do feel free to watch this or any of our on-demand webinars when it's convenient. Thanks again, and have a great rest of your day.